And what Martine asked me to do is just to sort of talk a little bit about trials. And to do that, what I'm going to do is I'm going to talk to you a little bit about the things that we're doing in Birmingham. Let's check this. Oh, that works. Good. So that you understand a little bit about, what we're up to. So you heard we run a dedicated clinic on a Wednesday afternoon. It's quite busy now. And we've also got UK PSC, which most of you have contributed in terms of genetics to, which is now properly funded and run from Birmingham, but it's a national study about PSC. And the reason that we're all here is that there's still a lot of things that we need to do for our patients. Okay, so and the slides are here. You don't have to understand everything on the slides. It's just to make you think about the reasons why we think it's important, and is to reiterate some of the things that Dave said. So this is the 20-year history of liver transplantation in the United Kingdom. And this is the percentage of patients who get, out of all the patients having a transplant, who will have a particular disease. So we've done pretty well with PBC, that related disease, that over time the number of patients needing a transplant has gone down. It may even still be going down. But you can see that for patients with PSC, there's been no budge at all in the rate of patients needing transplantation. So we really have to make inroads into getting new treatments. And to get new treatments, it is complicated, and there's a pathway, and we can't change that pathway. That's regulated, legislated, and it also interacts with commercial interests. And Dave sort of told you some of the things that you're going to want to have if you're going to have a better treatment for this disease. And you've got those three things that you're all worried about, which make it complicated. First of all, it's patchy. There are blockages, okay? There's inflammation, and there's cancer risk. And they don't all need to be there at the same time. And if we want the best drug, then we're going to have to overcome those. And some drugs may do better for some things, and some drugs may do better for other things. So we've got drugs that we'd like to be around inflammation, drugs around the scarring, and then we've got this whole issue of the cancer risk, which does make PSC stand out compared to other autoimmune diseases. You don't really see that in any other autoimmune disease apart from PSC. And you kind of understand what cirrhosis is. Cirrhosis is your scarred liver, okay? And some of the concepts that have been talked about for PSC is people have talked about calling it cirrhosis of the bile ducts. Okay, now it isn't cirrhosis of the bile ducts, but it's a good way to think about it, because that's your little bile duct in the middle. And when the disease has gone through its inflammatory stage, it's surrounded by scar tissue. And that's why the bile duct is blocked. That's what's happening inside you, and it's happening in the larger bile ducts rather than the smaller bile ducts. When you see someone with PBC, you don't see scar tissue around their bile duct. It's totally different. Okay? And you can see, therefore, that our challenge for our treatment is we really probably want to get to treat you before you've got all that scar tissue, because you'll know there aren't very good treatment for burns. I mean, burns doctors don't get rid of patients' burns with any of their medicines. So it's better to start earlier than wait till you've got the scar tissue. Okay? This is the list of prior treatments. You don't need to read it, okay? We wrote something for one of the medical journals, and they're so uninterested in the prior treatments because they haven't worked, they told us to put it in the supplementary file, which is because they don't think anyone will read them. But it's quite a long list of things that everyone has tried to work. So people have tried, and it's interesting. Dave shot off. Yesterday I was in Woburn with Roger Chapman, and we were teaching the the doctors in that kind of neighbourhood, Luton, Milton Keynes, that Stoke Mandeville, those kind of consultants came to listen to us talk. And when Roger started, it was in 1976, okay, and it was at the Royal Free, and the patients that he saw were very, very different to what we see nowadays, because he was only seeing patients at the very end. But over that time, they've tried everything, okay, everything logical and everything they can get their hand on. Now, Urso, which you'll all know about, is the most studied of the drugs, okay, but I want you to take note that if you actually look at the number of people studied, it's very small, okay? Some of the studies were 6, 20, 100 people. The biggest study, 150. Well, in the two current studies that are going on, the norurso study and the Gilead study, both of those studies are bigger than what was considered the largest study in 2009. And that's what you need to really get to the answer, because, we've alluded to the fact that you're all slightly different. Okay, and so you need to have reasonable sized studies to actually get a real answer. So there's a challenge there that even in the drug that many of you may be taking, you can count up that maybe only 400 or 500 people were actually in clinical trials to see whether it worked. You know, remember when they worked out that aspirin worked in a heart attack? You know, those studies had 20,000 people in it, if not more. Okay, you know, and you develop a drug for high blood pressure, you will have a study of, you know, 50,000 people. Okay, that's, you know, dwarfs that. Now, it's obviously more common, but it tells you why you can be sure. I tell all my patients this, not because I know which trial will work, but I know two things. The easy thing is the NHS will not pay for a new drug that is not proven to work. Okay, that is not how it is anymore. Okay, it may have been different as things were evolving 30, 40 years ago, but we cannot write any drug that we like. Okay, and the likelihood is that any drug that works will be costed at £10,000 a year. So they're not just going to do that. We have to prove it, which means trials. And the second thing is if you don't do the trials, you don't get the right answers. Okay, so everyone said UDCA was great for PSC, great for PBC. The problem in PSC must be, well, it's large ducts, not small ducts. Just give more. Give more, you know. Surely it's just that it's just a simplistic problem. So they did give more, and it wasn't so good for the patients. Okay, but you only found out because the patients took part in the study and were followed up, and you got the answer. You know, it didn't answer everything. We don't know whether it helped some people, but we do know for sure that high-dose UDCA is a no-no. Okay, and we only found out because there's a study, and studies have regular visits and have rules. And that's how you can capture data carefully, because, you know, clinics are great, but that's, you know, you need even more intense interventions. So who needs new treatments? It's been touched upon by some of the questions. So, you know, what you want to ask me is, if you've got PSC, am I one of those people who's going to do well, or one of those people who may one day need a transplant? So remember, when they studied everybody in Holland who's got PSC, they discovered that if they only looked at the patients who went to centres like Birmingham, well, then the time to transplantation was 12.2 years. If they studied everyone outside of Birmingham, it was 22 years. Okay? So there were plenty of people who never got to the big centres who had a disease they didn't want, but were living an average of 20 years, which means that's the 50%. So remember, there'll be some who were living 30 years, 40 years. So you can see the real challenges of who needs a new treatment. Okay? And of course this comes up, and we've talked about it already, what's my risk of developing cancer? It's probably an independent risk unrelated to your disease progression. Okay? But that's something we haven't cracked. And what we're trying to do is personalised medicine. And that's what everyone talks about. We're not there yet, but we're starting to try and encourage our clinicians around the country, and we hope we do it in clinic ourselves, to say, what's the likelihood of something happening to you? What can I tell you from your blood test? What can I tell you from your MRI scan? What can I tell you from your ultrasound? What can I tell you from your story as to whether or not you'll do well or whether you'll do badly? And we touched upon a few things. I can tell you that if your disease is only in the small bile ducts, you'll do well. I can tell you that the group of patients who don't have overt colitis tend to do better. I can tell you that probably women with disease have a slightly milder course than men. Not always, but sometimes. Okay? I can tell you that your blood tests start to tell me whether you're at high risk or low risk. If you've got normal blood tests with PSC, it's not actually a surprise. You do quite well, because what determines what, you know, how you do in PSC is how much bile duct inflammation there is, and our blood tests are not bad at picking that up. Okay? So we're starting to try and get to so-called personalised medicines. And, you know, the question was at the back about, you know, what do we do with these blood tests? What we're trying to do, and this is Oxford data, single centre, you know, just to be careful, you know, whenever you see these graphs, you know, people alive or people not doing so well, the tail end you probably should try not to look at in your mind, okay? Because there's nobody being studied, okay? You've got nobody actually who's lived that long, or you've got one or two people. So you really should look at the first half of the graph. But this is what Roger showed, which is logical, comes from really PBC work, that your blood test, your alkaline phosphatase, if you had persistently less than 1.5 times the upper limit of normal, that's probably somewhere in the region of less than 200. Depends on the machine you use. You do quite well. If you've got no improvement, you know, you're going to run into more problems. You've got a higher chance of developing the need for transplant. And that's this idea that we're personalising to what happens. And this is some of the data that Liz has got, who's been looking through all your data from UK PSC, and she presented at EASL, one of our liver meetings, and it works when we start to get up to 1,000 patients, or, you know, there's 750 patients with PSC there. Okay, you know, it looks at what happens if your alkaline phosphatase is more than twice the upper limit normal or less than twice the upper limit normal, and not surprisingly, it changes what happens to you. That doesn't explain why it changed, why it tells you that, but it starts to help us work out who would benefit most from treatment, we think. okay, because this is in months. You can see that things happen down the line, but that tells you perhaps which direction you're headed. Where it becomes really hard, and again, don't worry about the details of this slide, okay? Where it becomes really hard is that Urso will drop your blood tests. Okay, we know that. It drops everyone's blood tests. But I showed you the trial didn't work, and this just shows you in another way that this was a very important study done from Scandinavia where they looked at Urso against no Urso, and you can see there's no difference between those two lines. And then you can see in both of these graphs there is a difference, okay? But it didn't matter, the difference between those groups had nothing to do with whether you took Urso. Okay, because basically you could predict it just on what your blood test did, regardless of whether your blood tests were low because of the Urso or because they were low because that's the disease you've got. Okay, so it starts to complicate our whole problem is that we've got a drug that drops your alk phos, but doesn't necessarily make you live longer, and that's because disease is complicated. So where are we headed? So this is clinic at the moment, and this is what will probably be in the future within the next couple of years in Birmingham and around the country, and James will talk about how clinics are going to evolve. And what we need to move for, it's a strange world that we live in the NHS. I can get, give me a couple of £1,000, I can sequence your entire DNA. That's easy, okay? If I try and ask every clinician in the country to tell me all your blood tests, to tell you how you're feeling, and to get a copy of your biopsy results, it's nearly impossible. Okay, yet all that information is there. We know it's there, and it all belongs to one person, the NHS, but it's just impenetrable because it's such a complex organisation. And that's what we want to move to. We want to move from where we just see you and do things, and it's just a letter to your GP and maybe you read it, to where we actually extract the information continuously, and James may mention this about using information technology, so we learn about things. So we hope that in the next year, our clinic will move to the Institute of Translational Medicine, at least in part. And when you come, in addition to seeing everyone and having your blood test done, you might fill in some questionnaires that get put on the computer. You'll probably do some blood tests that go in the freezer for research for the future. So that when someone rings it up and says, oh, I've got a new test to tell me what happens to PSC, have you got blood going back 5 years where you know exactly what happened to the patients? We can say, yes, we have. All this costs money, but it's not actually a huge thing. We're doing most of it already, so we're just tacking on. You might get an MRI, Dave talked about that. You might get a fibroscan, that's probably more useful and cheaper. Okay, but we'll write down the result. And the letter, we hope, will be much more about you and what's going to happen to you than it is just about PSC and what PSC says in the textbook, which of course is one of the things that we think is important about specialist clinics, that when you go to non-specialist practitioners, you often just get what they've read in the book rather than what applies to you. Okay, and then we hope there'll be treatments which will start off as trials, but will eventually be hopefully mainstay treatment. And we hope that patients will be followed in a much more proactive way, where something happens, we react to the test that we have. We don't just make a diagnosis and say, oh well, let's just wait till you need your new liver. Okay, we actually try and change it before it gets there. It's a long way to go, and it's not easy because it's a rare disease, but that's the goal. And you see you'll be put into low risk or high risk. The high risk will skip to trial straight away. The low risk, and everyone will have the treatment that we have at the moment, and we'll continually try and say, what's your risk? Okay, so what should I do differently? So the problem is that, when you think about PSC, PBC, in fact, when you think about any disease, it's usually pretty obvious to think about how you'd like to treat it. And then when you try it, it doesn't work, or it doesn't work well enough. Okay? And that's where we've been a lot with bile duct diseases. You'd think it'd be obvious what to do, but it hasn't quite worked out that way. And some of the things that have changed, and again, ignore all the little pretty pictures, I do. The bile is not just a detergent. There are billion-dollar businesses now based on understanding bile. Okay, so bile is not just a detergent. You do need it to, absorb your fat, but actually it's really clever, and it's a really very complicated pathway that goes back right to the beginnings of evolution. How you control bile acid signaling from liver cells, bile ducts, the bowel. We talked about the bowel a bit, and back again. Remember, 95% of your bile acids are recirculated and reused. That's a pretty efficient environmental system, okay? And these are all things that are involved in inflammation, involved with how you use insulin, everything, okay? So suddenly it's become a target we can actually attack. The second thing is that tube that is causing all the problems, your bile ducts, is actually not just sitting there as a tube channeling everything out. If you were designing your bile ducts, you too would say to yourself, God, this is a bit unpleasant. I am sitting here and you're pouring caustic detergent down me all the time, which is designed to, it's meant to get rid of all your toxins. I don't like this. How am I going to stop myself from getting blocked? And it does. And what it does, for anyone, the chemists in the room, it's quite clever really. It pumps out bicarbonate. Okay, so that it's like an antidote to the bile acid. Okay, it's not. Most things in medicine are actually very simple. Okay, and that's quite a simple concept, isn't it? You would want more bicarbonate if you've got acid pouring down you all the time. It neutralizes it, but during the disease, it probably loses this umbrella, and that can contribute to the disease. And then on top of that, we might be able to get drugs, and norurso is one drug, which is pumped out across the large bile ducts and increases the amount of bicarbonate and is hypothesized to make the bile duct more resistant to damage, which may be better. So it helps us think about therapy. And the last thing is, as Dave said, it is all about your bowel. It's amazing what you can do to a mouse by fiddling with their gut microbiome. Okay, you can make it fat, you can make it thin, you can make it have diabetes. You can do all sorts by changing, what's going on in the bowel. So it's not everything. But one of the reasons why autoimmunity is probably increasing, and certainly, for those of you with inflammatory bowel disease, you'll know that if there's a plague of inflammatory bowel disease. The rates of inflammatory bowel disease in children is going up significantly. In parts of Toronto, Canada, it's really significant increases because our diets have changed much quicker than our genes. So over the last 100 years, the way we live is very different to the last 20,000 years that designed ourselves. Okay, but we talk to each other from the bowel through the signaling molecules. That it's always talking, and it's actually a two-way conversation. Okay, so we want to prolong quality and quantity of life. That would be our goal. It's obviously easier said than done. We might want better bile acids, a better urso, better drugs to turn off the immune system. This is an autoimmune, auto-inflammatory disease, but as Dave said, all the classic drugs haven't helped us yet. Okay? Or we may go for drugs to stop fibrosis. And so the study that's going on at the moment, one of the biggest studies ever done in PSC, in fact, is Gilead's done a study of a drug that blocks fibrosis. But it's going to take us 2-3 years to find out whether it works. But there are over 250 patients around the world injecting themselves with this medicine to reduce to see whether it reduces scarring. Okay? Because they've taken the approach, well, maybe we can't solve the actual injury, but we do know that most people who need a liver transplant with PSC need it because of cirrhosis. So if they have less cirrhosis, they'll live longer. So that's their approach. Okay? We would like to have better treatments. Okay? These are terrible symptoms. Okay? Fatigue is a terrible symptom, but I would argue that I think itch is worse. Okay? Anyone who has had terrible pruritus, terrible itch from PSC or PBC will tell you how awful it is to have ants under your skin. Okay? And it's, people have been driven to suicide. It's unusual, obviously, thankfully. Okay? But the reason why itch is so terrible and the reason why itch relates to fatigue is you don't sleep. Okay, so if you don't sleep, you're going to be tired. Okay, and it becomes like a cycle of, torture. Okay, unfortunately, actually, clinicians are not that good at treating itch. There are lots of patients who come to us who have never had any treatment for their itch, and there are simple things we can do which can make immediate benefits, and they just don't get asked in clinic how they feel. It's just in and out. What's your blood test doing, and no treatment for the itch, and it's disappointing. But our goal is to improve all those things. Probably to treat fatigue, we'll have to treat the disease better. Okay, itch, there are other ideas. Okay, so we'll come on to immunotherapy. We know the fancy drugs, but what the industry are interested in, there's a whole list of people that are thinking about drugs for PSC and PBC to make your bile thinner, to make your bile less toxic. Okay, so that's quite exciting. Okay, and I'm going to talk about a few of these drugs just so you understand what we're doing. So this is the study that's going on at the moment. This is a drug made by Falk Pharma. It goes back to a guy called Michael Trauner, who then also goes to a really amazing bile acid physiologist in San Diego, who's been around for like 60 years working on this. And this is this bile acid, it's light urso, that basically, soothes your bile ducts. And it does a good trick in the mice. Okay, so we've given it to our patients. Okay. Patients got placebo, some people got a different dose, Okay, we don't know the answer yet, but that study is fully recruited. So that in itself is an achievement across all of Europe, UK. Okay, it's fully recruited, and we should have some first headline news within the next, say, 6 months, I think, from understanding from the company. And that'll be exciting. But it's difficult because that study, for you to think about it, was, only asked the patients in the study to give a blood test. Okay? But we just heard that PSC fluctuates. Okay? You can see why the patients and the company would rather just do the blood test. It's a lot cheaper and easier. But you've then got the problem, and why you need a big study, that people fluctuate all the time. So how are you going to tell what the fluctuation is and what's the treatment? Okay? And we just have to hope that there's not too much fluctuation and enough effect that we can persuade them to go on to do a bigger study. Okay? This is a drug that may come to PSC, and it's worth talking about because it's going to probably come to PBC. Okay? And there is a study in the US, but unfortunately in Europe the study is not going ahead because for some reason European Union regulators put some blocks to it, which was a bit disappointing that the company couldn't overcome. But basically, Urso is made from your own bile acid, chenodeoxycholic acid. It's a derivative, you make it in your bowel. And this drug is a chemical modification of chenodeoxycholic acid. And the reason we think it might come to PSC, at least maybe get to trial stage, is that when I give it to patients with PBC, their alkaline phosphatase drops. Significant falls. Okay? And you can see that within 2 weeks, again, you don't need to worry about the details, but this is what happens to a PBC patient who's getting just sugar tablets. Nothing happens to the alkaline phosphatase. This is what happens to the patients on the drug. You can see it goes down. Okay? So it's no surprise the company would like to try in PSC, and they are doing the study in the US, and so we'll see what that means for patients in Europe. Okay, so we'll talk about Dave's hypothesis and the last couple of slides. Okay, so he told you that one of the ideas about why you get PSC is that the sticky white cells that should stick to your colon start sticking to your bile ducts. Okay, and that then tells you, well, if you're going to make a difference to your patients, maybe we should just stop them sticking in the bile ducts. And if we stop them sticking in the bile ducts, that'll be a good start. Okay, and what Dave's done a lot of work on, and there are other groups have contributed, is understanding the different molecules. You can see all these different names of different molecules that are basically expressed in the wrong place. If you know the name and then you know what it looks like and the structure, then a company will make something that blocks it. They're all quite clever nowadays. You can make an antibody to anything. Okay, and that's essentially what's happening, and that's why we hope there'll be trials. So what we've got, Dave's too modest to call it his own idea, but it is his idea, okay, is the opportunity to block these molecules, and we hope therefore stop sticky cells from sticking in the wrong place. So we've opened our study of anti-VAT1. That's an adhesion molecule and also an enzyme that upregulates sticky molecules. And we haven't dosed our first patient, but we've started to screen. It's not easy. We want all our patients to have abnormal liver tests, and they've got to be prepared to take an infusion. But if we don't do the study, we'll never know. There comes a point where you have to move from the laboratory into to the patient. Otherwise, there is no further place to go. You can just keep having ideas. And then we're waiting for Takeda. So some of you who've got Crohn's will have heard of a drug called Vedolizumab. That's now licensed for Crohn's disease, and that blocks sticky white cells. And they should be launching a clinical trial, they've been saying this for 10 years, but probably 2016, of a drug that blocks that particular molecule. Okay? So that's quite exciting, because then you might have a drug that treats the colitis, and we can see whether or not it treats the inflammation in the liver. We don't know yet, time will tell. The other trial we hope to start next year in Birmingham and in other sites sort of reasonably close by is that with Phil Newsom we've got money from the European Union, whilst we're still members, okay, to give a form of, it's not really stem cells, it's something called stromal cells. So this is a more simplistic approach, although it's quite complicated to deliver, but a more simplistic approach to the disease, which says this disease is inflammatory. I know that when I infuse these particular cells into someone, in a mouse and in humans, they act like anti-inflammatory sponges. Let's see what happens when we give it to our patients with PSC. In a sense, it bypasses the complexities of the disease, which is so complex, has got so many different causes, and says, well, let's just forget about that. Okay, let's find a sponge that is anti-inflammatory that can sit there and soak up the bad things and maybe make a difference. So we hope to open that study sometime next year. It is a tour de force, however, to get permission to do this. Okay, do not underestimate, MHRA in licensing it is taking up a lot of our thinking at the moment. Okay, so a few things from your perspective. Trials are about safety, okay? And then we'd like to see whether things work. But everyone's got to be the same in the trial. If you're all different, we'll never prove something works. And that's why trials have rule books, and that's why they're so hard to set up, and why you have a nurse running trials who understands rules. Because you have to have reasons to get in the study and reasons why you might not go in the study. And it may be that you're screened for one study and it's not acceptable. That's nothing a reflection on you. That's the rules are the rules. And when we design the studies, we want everyone to be as close to the same as possible, because that makes it easier that we might even prove why, whether it works or not. So you've got to usually have PSC. We've got to know how active it is and where it is. We need to know whether you've got colitis. We need to know what you've done with your medicines now and in the past. It may be you need to be off Urso. It may be you can stop it. It may be you can stay on it. But we need to know, because it may change the blood tests. And we do, generally speaking, need to make sure no one gets pregnant, okay? Because drug companies don't like the idea of drugs and babies until, that scares them significantly, okay? Why might we exclude someone? Well, fundamentally, you've got to consent, okay? The documents are written not by us. We know how to write the consent form, but, there's a whole process. There's a reason they're 25 pages long. That's not our choice. Okay, but they are there in great detail for all sorts of obvious reasons, okay? Because for the majority of people taking part in a clinical trial, you may not gain yourself. We hope you will, and one day there's bound to be the study that is the wonder drug, but if it was that easy, then all pharmaceutical companies would be the size of GSK, and they're not, are they? Okay, and even the likes of GSK and Pfizer struggle to get new drugs, and they'll have, 20 drugs in phase 3, which only 1 or 2 might become their blockbuster. Okay, so you can see how hard it is, and so you've got to understand that. Generally, our trials are with patients where the liver works. Generally, we're not doing studies with patients with cancer, although if it was a study about cancer, that'd be different. Most of our studies are not to do with transplantation. Okay, we don't want you to start new medicines in the middle of the study, and we don't want you to stop old medicines without telling us, because that will just screw the whole thing up. Okay, and then we won't know the answer. Okay. And, you can see how, some of the studies are bigger, but, if you've got a study of 45, you can start to see how if one person starts a study then doesn't finish it for whatever reason, or starts a new medicine in the middle of it, your numbers start to go down, and you start to, makes it harder. So it's just the real world. And generally, you need to be otherwise well. How do we prove a drug works? Well, these are some of the statements that we're going to publish shortly from the International PSC Study Group, which are there to make you think about why studies are the way they are and what you might have to give up to take part in a study. Okay? In an early phase study, your blood test may be good enough. In the first phase of design, if it drops the alkaline phosphatase, that's a good signal, isn't it? It's not good enough, but that's what you want before you spend the next 50 million. Okay? But it's not going to be good enough to get you a new drug and get the government to pay for it. We said that biopsy is actually quite a good thing to think about. We know that you don't have biopsies during your normal care. Why would you? Generally, you don't need to, except for overlap. Okay? But actually, liver biopsy is where the disease is. It is a pretty solid marker of how much scar tissue there is, and you can see the inflammation. So you can see why some clinical trials are doing biopsies, because it's hard endpoint. If your biopsy gets better and the scar tissue goes away, that's going to be quite hard for people to say not to pay for that drug. Okay, if it's just your blood test, they'll say, well, Urso dropped your blood test, why is this any different to Urso? I'm not going to pay. Okay, and probably the next drug will need to use a mixture of different things. Probably a mixture of blood tests, maybe a biopsy, maybe a fibroscan, maybe an MRI. And we'll use lots of different things, because we can't wait forever to need transplant. Okay, so the last slide again looks pretty, but it's pretty simple actually. For anyone in the pharmaceutical industry who wants to tackle liver disease, they're thinking around, do I get a drug that makes the liver cell better? Do I get a drug that makes the immune system better? Do I get a drug that makes the bile ducts more stringent, more tolerant to what's going on? Or do I fiddle with your bowel, change the bugs, like some people take antibiotics, and see whether that helps the liver? Or do I have a drug, they'd like a drug that has one dominant effect but does a bit on all the others. Okay, I think that's all I have to say.